Hello, and in this video, we're going to be going over chapter 4, and we're going to talk about some important lab media that we're going to be using in upcoming labs, such as 1.1 and 1.2, and we're just going to do some simple testing for your first exam. So to start off with, we're going to discuss culture media. And so lots of prokaryotes have actually not been grown in culture media, but what we're going to use in lab, like I said, we'll talk about general purpose media, which is a tryptic soy agar. Then we'll use that in, for instance, lab 1.1 and 1.2. General purpose media allows for growth of lots of different types of microorganisms. It's rather generic, so it allows for growth of E. coli, staph species. We'll talk about Pseudomonas species that we can grow on as well. Next, we have selective media, and this is media that will allow growth of certain organisms while inhibiting growth of others. So it's very selective in what it wants to grow. The differential media allows for lots of different types of growth to take place, but it allows you to see differences in the microorganisms that are grown on the same plate. And then finally, we have some media that can be both selective and differential. So to start off with that general purpose media, again, this is able to grow a lot of different types of lab organisms, and we will really commonly be using this in lab. What's so nice about these general purpose medias, such as what we see down below as these tryptic soy agar plates or TSA plates, and they allow us to see colony morphologies. So in figure 1, what we can notice is that we have these little clusters, these little spherical clusters, and these are colonies. So we can see a macroscopic view of these organisms, and they're going to have a specific color. Their margins will look a certain way. Some may even be elevated, and this allows us to not only see them from a macroscopic view to be able to determine color, it also will allow us to do some key identification when it comes to the microorganism itself. And then we see this picture over with the handprint, and you can see all the different types of organisms that are growing on this plate. Some are going to be like a yellowish color, some are an orange color, some are like a cream beigey color, while others are white. Some look more like filamentous, some are going to have like an opaque look to them. And all of those different types of organisms are going to be a different type or different species of organism that we could potentially study. So it's a really cool picture to see. All right, so then moving on to selective media. This will inhibit growth of some bacteria while allowing for growth of others. So maybe this allows for growth of staph species, but it doesn't allow for growth of gram-negative bacteria. On the flip side, it may allow for growth of gram-negative bacteria while inhibiting the growth of gram-positive bacteria. So it's selective in what is able to grow. So here's just sort of a picture here that you can see. Again, looking at these different plates, we can see these bacterial colonies and we can see the growth that's taking place at a pH of 7.3. However, if we change the pH at 5.6, we notice that we don't have the same type of bacterial colonies growing, but rather we can see some fungal colonies growing instead, because certain microorganisms can only withstand and tolerate a specific pH, and that's determining how acidic or how basic these environments can be. Again, we can have different types of differential media, and this allows us to see differences in organisms that can grow on the same plate. So one example are blood agar plates. So they're going to have sheep's blood put within them, and we can see their differences in how they're able to break down red blood cells. So there's alpha hemolytic that doesn't have a complete lysis or breakdown of the red blood cell. So you may see this green zone that appears. In beta hemolytic, it completely lyses the red blood cell. So you see like a clear zone. And then with gamma hemolytic, there's actually not a lysis. So you won't see any change to the media itself. And that's what we can see on this picture here. We can see those differences between alpha, beta, and gamma. So again, you may see like a greenish hue. You can see that sort of like clear coloring, that there's really no difference to the actual media. And then again, we can see that halo or that complete lysis happening between alpha, beta, and gamma. All right, And again, some media will be both. It will be selective and differential. So when we talk about the mannitol salt agar plates, this is selective for organisms that can handle high salt tolerance. So for instance, a Staphylococcus species can actually grow very well in a mannitol salt agar plate. Usually high salt concentrations will inhibit most bacteria, so a lot of things won't be able to grow. But then it also allows for organisms to show up differently on the plate because mannitol is a sugar, so not all microbes can utilize this sugar. And there's also a pH indicator within the plate as well that allows the media to turn yellow when this acidic product is produced. So not only is it selective, but it allows for differences to be seen, especially with Staphylococcus species. So again, for the mannitol salt agar plate, again, lots of organisms are not going to be able to grow because they won't be able to withstand that high salt concentration. But on the plate down below, what we can see is this highlighting of a yellowish color. That highlighted yellow color is actually showing Staphylococcus aureus species. While in the more of the like white colony or more of a like a creamier color that you can see before, that may be an indication of a Micrococcus species or a Staphylococcus epidermidis species. More than likely, probably a Staphylococcus epidermidis species. We have what's called an EMB plate, an eosin methylene blue plate. This is selective for gram-negative coliforms. So again, gram-positive species will not really be able to grow on this plate because eosin and methylene blue actually inhibit gram-positive organisms. But we can also see a pH indicator as well for gram-negative bacteria. And so, two main things that we'll be able to see are E. coli, and E. coli will be able to give off this like greenish color when we are actually looking at the macroscopic view or presentation. So down below, you can see this E. coli in this sort of like metallic green color. So not only again is it selective, but it allows us to see those differences versus other gram-negative coliforms that may highlight like a purplish or like a much darker color. And then we'll do some biochemical testing as well. So every bacterial species produces different enzymes, and these are going to allow them to produce different byproducts as well through metabolism. So that means that not all bacteria are created equally, and they don't all metabolize the same way. So for instance, not all bacteria can metabolize lactose or sucrose or dextrose. So because of that, we can do biochemical testing and have some positive controls, and we can also have some negative controls as well. For instance, like the Simmons citrate test. This allows us to see an indication of whether or not the microorganism can utilize citrate. On the left-hand side, you're going to see a positive if it's blue, and on the right, you would see a negative if it's green. All right, so those are just some examples that we will discuss. We'll get into more detail about laboratory media and different lab techniques coming up very soon.